Sperm disorientation in space. New research highlights reproduction challenges for deep space travel. Human and animal reproduction in space may face significant hurdles, according to new research from the University of Adelaide, which shows sperm struggle to navigate in microgravity. The study, published in Communications Biology, demonstrates that sperm exposed to weightlessness become disoriented, potentially reducing fertilization rates. This finding has critical implications for long-term space settlements and interplanetary missions, including planned human habitats on Mars and the Moon. The challenge of sperm navigation in zero-gravity. Researchers used a 3D clinostat, a device that simulates microgravity by continuously rotating samples, to observe sperm from humans, mice, and pigs. The results were clear. Sperm exposed to simulated zero-gravity exhibited a 40% reduction in successful maze navigation compared to control groups. This disorientation occurs because sperm rely on gravity to orient themselves when traveling toward an egg. Without that reference point, they essentially tumble randomly, hindering their ability to reach their target. The study underscores a fundamental biological challenge for space colonization: the increasing focus on establishing permanent human presence beyond Earth, driven by projects like NASA's Artemis mission, SpaceX's Mars ambitions, and the rise of space resource extraction, necessitates understanding how basic biological processes function in extraterrestrial environments. The current findings suggest that reproduction in space may not be as simple as simply bringing humans and animals off-world. Beyond disorientation, embryonic development and radiation risks the challenges extend beyond sperm navigation. The Adelaide team also observed that microgravity affected embryonic development in pigs and mice, though healthy embryos still formed. Researchers believe that adding progesterone, a hormone naturally released by eggs, can help mitigate disorientation, as it may provide a chemical guidance cue for sperm. However, additional factors complicate the picture. Astronauts are also exposed to dangerous levels of radiation outside Earth's atmosphere, which damages sperm DNA and reduces fertility. Previous studies, dating back to the 1980s with experiments on rats in space, have already shown negative effects on testicular mass and embryonic development under similar conditions. NASA continues to run reproductive biology programs, including sending human sperm to the International Space Station to study the impacts of weightlessness. What this means for the future of space colonization, the research is not just theoretical. Elon Musk's reported, though denied, interest in seeding a Mars colony with his own sperm illustrates the practical urgency of this field. While the idea may sound like science fiction, the underlying science is real, and the implications are profound. As we progress toward becoming a spacefaring or multiplanetary species, understanding how microgravity affects the earliest stages of reproduction is critical, says John Colton, director of the University of Adelaide's Andy Thomas Center for Space Resources. Scientists are now calling for increased international collaboration to address knowledge gaps and establish ethical guidelines for reproduction in space. The next steps include studying the effects of varying gravitational forces on the Moon and Mars, as well as developing countermeasures to protect sperm from radiation. Ultimately, solving these biological puzzles is essential for ensuring that humanity can not only reach for the stars, but also build sustainable, self-sufficient settlements beyond Earth.